Hi, my name is David Hughes and I'm a statistician at the University of Liverpool. I work in the Department of Biostatistics and my main research interest is into methods of clinical classification. All around us, huge amounts of data are being collected. That's certainly true in a medical application, which is my main research interest. Every time a patient visits a hospital, an abundance of data is collected about them. If the patient has a known condition or is thought to be at risk of developing a particular disease, then doctors might collect information from blood samples like cholesterol levels, hemoglobin levels, or blood pressure, or they might collect information from various medical images like X-rays, MRI scans, or retinal scans. Statistics is the process by which we transform data into knowledge. The question I'm most interested in is how can we use this data to identify patients who are at high risk of either developing a disease or requiring an intervention. This has a number of useful benefits, which I'll talk about later, but essentially early identification of patients who are at high risk might lead to early intervention in their treatment, which could improve patient health. The problem is, analysing the abundance of clinical data available isn't easy. A patient's data is complicated for a number of reasons. Observations of a variable collected repeatedly over time are certainly likely to be correlated in some way. In addition, a patient's blood pressure is likely to be related to their cholesterol somehow. Most statistical methods are only designed to analyse a single variable collected over time, but being able to incorporate information about additional variables might increase our ability to accurately identify patients at risk of disease. The main part of my research has been to develop new statistical techniques to analyse the complex longitudinal data I've been describing. Along with my colleagues, we've developed methods of longitudinal discriminant analysis and also developed computer software in the R language to implement our methods. We're now able to analyse multiple longitudinal variables of different types and to use them to classify patients into clinically relevant prognostic groups. Although I'm a statistician, my time is not all spent with complicated equations. Our main reason for developing these methods is so that we can analyse medical data and provide a benefit for people. So far, we've been applying our statistical methods in two clinical areas. The first is in the field of epilepsy. Epilepsy is a serious neurological condition that affects over 50 million people worldwide. Unfortunately, around 30% of patients with epilepsy will not achieve remission from seizures, even when given anti-epileptic medication. We have used our longitudinal discriminant analysis methods to model the seizure history and treatment history of patients in order to identify those patients who will not achieve remission from seizures. Our initial findings are promising, since we can correctly identify over 90% of patients with drug-resistant epilepsy, and in addition, in most cases we're able to identify these patients between 2 and 3 years earlier than is currently possible. In addition, we've shown that our models work well on data from 2 additional clinical epilepsy studies. This is a crucial first step in demonstrating the usefulness of our model, which might be a first step in the early identification of patients with drug-resistant epilepsy. Our second application comes from patients with diabetes. Every year these patients attend clinics for a retinal screening to try and identify patients who are at risk of developing sight-threatening diabetic retinopathy, an eye disease that can lead to blindness. In addition to retinal screenings, various other information is collected about each patient, including their glycated hemoglobin levels, cholesterol, and blood pressure. We've been able to identify over 85% of patients who will develop sight-threatening diabetic retinopathy within the next year. Being able to accurately identify these patients will allow them to be monitored more closely and given appropriate treatment.